Okay, so picking back up with some diagnostic techniques, there's a special type of media that is called MGIT, which stands for Mycobacterial Growth Indicator 2. It detects growth of a slow-growing Mycobacterium tuberculosis bacteria. Some organisms are so fastidious in their growth patterns that we have had to develop specific and special media just for them because they grow so sporadically on other media. But this type of media monitors oxygen levels in a tube, which is inoculated with the patient's sputum or patient's blood. There is a silicone chip at the bottom of the tube that has been impregnated with a fluorescent substance sensitive to oxygen levels. It'll begin to fluoresce as the oxygen levels in the medium decrease. There will be automated machines which will give a visual and audible signal so a positive culture can be identified. So as the oxygen level depletes by this oxygen consuming organism, you will get a signal telling you the culture has begun to grow, and then we can identify it as mycobacterium tuberculosis. Uh, Antimicrobial susceptibility testing is useful in determining which drugs will be used in treatment. One of the tests we use in our lab is something called the Kirby-Bauer test. That is an example of a susceptibility test. Um, with the crisis level rise in antimicrobial resistance, it's important not to waste valuable treatment time by guessing about which drugs to prescribe. So in some cases, it's better to wait just a couple of more minutes to be sure that you're not treating with the wrong antibiotic. Most of the automated phenotypic systems incorporate antimicrobial susceptibility testing during their pathogen identification. This type of test is also very useful when you start to identify species such as the Streptococcus organisms, Clostridiums, such as C. diff, and the Pseudomonas organisms. Another type of test that is very active, very accurate, is something called phage typing. The bacteriophage are a group of viruses that only infect bacteria. And they infect these bacteria in a species-specific and strain-specific way, which is useful in identifying some bacteria. So not only are these things species-specific, they tell you it's E. coli, but they are also strain-specific. They tell you which strain of E. coli. Is it E. coli O150 or O1H157, whatever the exact number is? It is often used for tracing bacterial strains in epidemics. A lawn of bacterial cells is inoculated onto an agar plate. The plate is mapped off into blocks. The blocks are labeled, and then a particular phage type is exposed in each block. Cleared areas corresponding to lysosomal cells will indicate sensitivity to that phage, and susceptible animals needed to cultivate Mycobacterium leprae, for instance, and Treponema pallidum. These are two bacteria that are notoriously difficult to grow in the lab, and generally we have these growing leprosy. We grow that in armadillos, for instance. Avian embryos and cell cultures can be used to grow host cells. Dependent rickettsias, that's for the different Rocky Mountain spotted fever things, certain chlamydias, and certain viruses. Here's an example of the plate. Notice the little blocks that are set up. You've got a growth medium here. The entire lawn of this plate will be covered with the bacterial growth, and then the different phage types. Like this would be phage 79, and this would be phage 52A. This is phage 52. That looks like 29 right there. There's 3A, et cetera, et cetera. And you'll notice the only two that are showing any reaction to this particular microbe are phage 52A and phage 79. So, and then there is a little artist rendering of the phage types be going in and destroying the cells. You have even bacterial growth all over here, but you have this uneven no growth pattern here because of the phage typing. How do we determine, or what is the usefulness of determining the clinical significance of cultures? It is important in a diagnostic way to rapidly identify if an isolate from a specimen is clinically important, or is it merely a contaminant or normal microbiota. Example, a few colonies of E. coli in a urine sample can be indicative of normal microbiota, but several hundred can mean a natural infection or an active infection. A single colony of a true pathogen, such as Mycobacterium tuberculosis, in a sputum culture, or an opportunist in a sterile site, is highly suggestive of a disease. So you have a few E. coli in your urine, that's probably normal. You have several hundred, you probably have a UTI. However, the presence of a single Mycobacterium tuberculosis in a sputum culture is probably suggestive of disease. Repeated isolation of a relatively pure culture of a microorganism can mean it is an agent of disease. We can repeatedly, off of one sample, isolate a relatively pure culture of some microorganism. Could be, or can be, an indicator of disease. The drawbacks of phenotypic methods, when the microbe has to be cultured, it's going to take a minimum of 18, 20 to 24 hours, and oftentimes longer than that. And as I will tell you when you take a microbiology class with me, all the time you are doing these tests, what is your patient doing? Most common answer, getting sicker. I say your patient's dying. Your patient is dying during that 18 to 24 hours. Many infectious conditions may be caused by non-cultural organisms, which will leave open the possibility that the organism that we do culture is simply a bystander. It's very possible that we've cultured something that looks like the causative agent, but it may actually be that the causative agent is something that we can't grow in the lab. Phenotypic diagnostic methods, again, direct examination, microscopy of patient specimens, usually after staining. Things that require cultivation, biochemical testing, susceptibility testing, selective and differential media, all of those miscellaneous tests we just talked about, all of those things require cultivation. All of them take multiple hours and days to accomplish. And what is your patient doing while you're doing all the testing? Your patient is dying. Serology tests involve the in vitro testing of serum, urine, cerebrospinal fluid, tissues, or saliva for the presence of specific antibodies. Based on the principle that antibodies have an extreme specificity for antigens, therefore their presence will reveal the history of a patient's contact with microorganisms or other antigens. In other words, if you are trying to look for specific antibodies and you find them, that means at some point that patient has been exposed to the antigens. We can do these several different ways. You can visualize the interaction of antigens and antibodies macroscopically. If you've ever seen blood clotting, you've done a little simple blood test where you're trying to determine if you were type A or type B. That is an agglutination reaction. That is a visualization of the antigen-antibody complex. You can also look microscopically, but whether you're doing it macroscopically or microscopically, either one is a powerful tool for detecting, identifying, and quantifying these antigen-antibody reactions. This type of test is used to determine the immunological status of patients, to confirm a suspected diagnosis, and to screen individuals for different diseases. And here's a couple of examples of different types of serological testing. You take a sample, you put it in with the known antigens. If there is no antigen complex here, then you get no reaction. But here you see the antigen and the antibody. They are coming together here, so you get a reaction shown over here as the agglutination reactions. Same things over here. You get the bacteria from a growing colony. You put in the antiserum for Staph aureus. If you get a reaction, that means the bacteria growing is Staph aureus. If you do not get the reaction, then it is not Staph aureus. Different kinds of antigen-antibody interactions. A molecular basis of an immunological test is the binding of the antigen and the antibody. It's the binding of the antibody to a specific site or epitope of the antigen. Binding reaction is not always visible to the naked eye, nor in light microscopy. If the antigen-antibody complex is suspended in a solution, under the right conditions, binding can be seen as clumps in the solution. They'll sort of settle out, and you'll have a little sediment at the bottom of your tube. Agglutination and precipitation reactions are types of reactions where you do have visual confirmation of your test. In both of these reactions, one antigen is linked by several antibodies to form insoluble aggregates that will settle out. An agglutination reaction is where antigens are whole cells or organisms such as red blood cells, bacteria, or viruses. They will form visible clumps. The Wheel-Felix reaction diagnoses rickettsial infections, for instance. These are also used to determine blood compatibility. Precipitation reaction is where an antigen is examined in a soluble molecule. These are more difficult to visualize because the precipitates are easily disrupted in liquid media. You have a layer at the bottom and you pick your tube up. By the time you pick your tube up, get it up to eye level, you may have disrupted it enough that they've sort of gone back into solution again. Here's a couple of different types of tests, agglutination tests. This is a typical sort of blood test well. When we do the simulated AIDS test on in our labs on this campus, this is the type of thing we're looking for. We're using one of these 96 well plates and doing looking for this complex to either form and settle out or not to form. And then over here is what is referred to as a serial dilution. Immunochromatography is a lateral flow test found in drugstore pregnancy tests and rapid strep tests in your doctor's office. You have a plastic cartilage that contains a porous material or a polymer that directs the fluid to flow in a particular direction. The fluid, during a pregnancy test, the urine, will encounter antibodies along its route of flow. If the sample contains the correct antigen, it will bind to the antibodies and continue on to the next station in the cartridge. The next stage will contain a third molecule that is impregnated on the paper in a striped pattern, and you wind up with the control telling you the experiment worked, and here's the test. The third molecule will bind the complexes, and eventually this causes the stripe to change color. In this way, a microscopic antigen-antibody positive reaction is made visible to the naked eye. Again, put your PP sample down here. The direction of flow is this way. Here's your test. Here's the control. This one is going to change color just to indicate that whatever you put here has flowed down to here. So one blue bar, not pregnant, two blue bars, pregnant. Antibody titers, this is where you are looking for the dilution or the percentage or the concentration of antibodies in a sample. This is determined by serially diluting the patient's serum into test tubes or wells on a microtiter plate, which contains equal amounts of bacterial cells or the antigen. The reflection of the highest dilution of the serum that still produces an agglutination. The more a serial serum sample can be diluted and still react with the antigen, the greater the concentration of antibodies, and thus its titer. And this is used to diagnose autoimmune disorders and to determine past exposures to certain diseases. Serotyping is an antigen-antibody technique for identifying, classifying, and subgrouping certain bacteria into categories called serotypes. It employs antisera against cell antigens such as a capsule, a flagella, and a cell wall. It is widely used in identifying Salmonella species and Salmonella strains, and it is the basis for differentiating pneumococcal and streptococcal serotypes. The Western blot technique or Western blot procedure is a test done as a second or a verification test in some tests, for instance, like the HIV/AIDS test. We will perform an ELISA first, and then if the ELISA is positive, we will then move on to the Western blot. Proteins within the cell will lysate. Cell lysates are separated by electrical charge within a gel. The proteins in the gel are transferred to a special filter. The filter is then incubated with specific antibody solutions labeled with radioactive, fluorescent, or luminescent molecules. Sites of specimen antigen-antibody binding will appear as a pattern of bands that can be compared to a known positive and negative controls. And this verifies microbial specific antigens or antibodies in a patient sample. And here is a Western blot. This is the test done here. This is an artist rendering of what you're having to have. This is the colorimetric label that's attached to the secondary antibody. This is the secondary antibody against the patient antibody or the primary antibody. Here's the membrane blot. There's the target protein, which is bound here. If the proper antigen is there, it will bind here, the secondary here. And if all of these things do not align, then you do not have a positive reaction to the Western blot, and you'll see a result more like this rather than this. All right, I'm going to stop there for a couple of minutes, give my throat a rest, and then I will come back and finish up in a few minutes.